Hello, and thanks for your interest in this session and presentation. I hope you benefit from participating. More important, I hope you are well and appreciating this opportunity to connect, although virtually. Kudos to ASHS and others for making that possible. And at the same time, let's hope for the opportunity to meet in person soon. Today, I am pleased to update you on work my team, collaborators, and I are completing that focuses on ultimately developing best practices, research-based recommendations for selecting, using, and evaluating microbe-containing biostimulants. A number of presentations at the conference focus on these products, so I hope the info I'm going to share complements them well. You may know that the most recent Farm Bill was the first bill to include official language detailing biostimulants. And this language opens the door to more effective communication and research regarding biostimulants, an important amount. The landscape of biostimulants is very large and diverse, as you know, with microbe-containing biostimulants just representing one sector of that landscape, one part of that landscape. And if we look very carefully at the microbial biostimulants specifically, we recognize that they contain bacteria, fungi, or both. And as their primary active ingredient, that's truly what sets them apart from other biostimulants. I would say that microbe-containing biostimulants are a bit of an enigma, if you will. They are abundant. There are dozens to hundreds available right now. They are increasingly popular, used by a large and increasing set of growers. They certainly generate a lot of revenue. Hundreds of millions of dollars are spent on them annually. But they're also ridiculed and somewhat disregarded because they have, as a class, if you will, a negative reputation and some accusations that are rooted in some fact. This perception is a challenge for farming industry and research extension communities, all of whom would like to, if you will, establish a quality baseline on the role that microbe-containing biostimulants can have going forward. Some of those challenges are often rooted in lingering questions, such as, what is their host specificity exactly? Under what soil and cropping conditions do they perform most adequately? What are the best application procedures? The 4 R's, so to speak: rate, timing, placement, amount, or location. Interactions with other inputs, especially pesticides. Can one apply a fungicide and a fungal-containing biostimulant at the same time, or even to the same crop? And by the way, once they're applied, how do we evaluate evaluate them effectively? The list of questions is large and continues to grow, actually. All of which point to the fact that selecting, using, evaluating, and then advising people on them, as we often do, can be very, very difficult. And establishing clear guidelines for the use on them and on their efficacy and on their returns on investment is equally challenging. And all of us as extension research folks, and even just research folks as well, and influencing, if you will, the thinking of others, would like to have very solid data to assist us in our work. I like to think that what we know about microbe-containing biostimulants is like this farm pond, and what we truly need and want to know is something much larger and something much more powerful. And moving from one to the other, from our current situation to where we'd like to go, like this barn raising, is very likely to involve a solid foundation or require a solid foundation, lots of teamwork, and a bit of a plan. I've been fortunate to work with the support and collaboration of a number of entities, and certainly the collaboration and support through USDA and other programs, the support of Tri-Ishtal, SARE, a USDA ORG program, and companies as well, and a list of really good, talented, bright people who know so much more than I do in certain areas, that really brings that needed expertise to the table and informs the work that can take place. Dr. Jung Wong now with UC Davis, Julie Laudick, currently Nicole, Sonia, and Mark as members of my team, who are truly responsible for the research-based numbers I'm going to be sharing with you today. With them, we've been able to complete on-farm evaluations. We've been able to complete on-station evaluations. We've been able to do so across 7 crops involving 13 products and as representing 10 different companies. We have completed essentially 11 experiments, totaling 39 runs. Each of those experiments involve 32 to 72 inoculated plots. You see the list of crops there and where they were grown and the types of settings in which they were grown. An individual experiment for the purposes of this discussion included 1 crop seeded or transplanted in spring, summer, or fall at a transitional or a certified organic site in an open field or a high tunnel setting, and the application of up to 6 products at either one of 3 crop development stages, a timing experiment, or percents of their recommended rates, a rate experiment. So the products used in the experiments are listed here, and their general makeup is also detailed in terms of their bacterial, fungal composition, number of genera, number of species. Charting out those 11 different experiments and 39 runs would look something like this, and certainly not an opportunity to go through this in detail. But if you were to pause the recording, you can examine more closely using this chart, which experiments were timing related, which were rate related, and what products were involved. These experiments were often large scale, especially in the open field. They were completed across 5 different crops, 2 in the field, 3 in high tunnels. They involved, as much as possible, the use of controlled but commercially representative settings, field and high tunnel, and procedures, much like you're being depicted here. And of course, careful sampling, like all of you are very familiar with. And inoculation was often completed by hand, which is of course tedious but very reliable. It was typically followed more or less immediately by irrigation, and in some cases it was actually replaced by injection in other experiments, injection into the irrigation line. We took what steps we could to record crop development throughout all of the experiments. Of course, recording yield per plot, examining crop quality as well, whether it be sensory or other parameters. And our priorities in these experiments, on the first was to identify macro responses to inoculation, plus or minus, the season, the setting, the product rate, timing, very important to us. Second priority was to develop analytical approaches for understanding the treatment effects and their underlying causes. And our third priority is to compare the outcomes from specific product by treatment combinations. I'd like to emphasize that these 11 experiments were not designed to identify the top performing product in much the same way that other experiments are and experiments and evaluations are. Along the way, for those types of experiments, we worked with individual companies or clusters of companies in more focused projects or experiments. In this case, grafted and ungrafted tomato being grown with Invigorate. In other cases, we integrated evaluations of microbial biostimulants with the performance of different types of grafted plants, varying in their rootstock scion combination and being planted in standard or plastic covered raised bed or in strip till systems. Again, a very large experiment unto itself, but again, combining grafting and microbial biostimulants, data from that experiment not to be discussed here. But again, pictures of it, another example of a large-scale experiment. And again, at the same time, more well-positioned to identify a relatively better performer from another, if you will, if there is to be one, than the 11 experiments I was detailing earlier. Our data analysis, which is something I'd like to spend a few more minutes on, has been and will continue to be descriptive, inferential, and parametric and non-parametric in nature. We, of course, are looking at a large group of yield and quality variables and plant variables. We have a large diversity of experiments in terms of the 5 different crops, the 2 different settings, and the transitional and certifiable organic settings. So a one-size-fits-all approach will not be appropriate. So we will be using and have continued to use a full battery of statistical approaches. And I just like to point out, or remind us all that reports in multiple literatures, including the horticulture literature, reference the value of multiple statistical approaches, less focused on means testing, which is somewhat common in product testing and other similar research. Just the main message being that the value of means testing is unquestioned overall, but its application in certain situations may be less than optimal. And we are openly wondering, I guess, within our team and collaboration team, if that is the case with the experiments that we've conducted. So I'd like to just summarize some preliminary results for you very quickly. In widespread trialing effects, of course, in the past, if you look at the literature and perhaps other experiments, work being described here at the conference, effects are notoriously case-specific, that is inconsistent, and yield increases are often rare, sometimes below 5%. And there are 3 categories of reasons for that. Product as it reaches the investigator may be subpar. The handling, storage, or application of that product may be inappropriate, and the evaluation protocol, whether it be the data collection or statistical approaches, may be inadequate for the purpose. In our case, showing here just an example of one experiment, springtime carrot root yield values by year, all products and all timing treatments, we're showing an example of our tests for normality in the data. And so far, most of those tests have come out positive, although ranges differ. You can see in this case, excuse me, that the distributions are relatively normal, but they do cover different ranges by year. I also like to draw your attention to a variable that we are now using, which is called the I to NI ratio. We have a control plot surrounded by plots that received a treatment in the same replicate, and each of those has a yield value, and we simply divide the yield value of the inoculated plot by the control plot for that rep and create an I over NI value for each plot. If it's greater than 1, the inoculation had a positive effect. If it's less than 1, inoculation had a negative effect. We can plot and track those I over NI values throughout the entire batch of our experiments. And note that in the high tunnel experiments, 54% of those values were greater than 1, and in the field experiments, 50% of them were greater than 1. We can then do a histogram of those I over NI values, for example here with the high tunnel ones, and see that they are somewhat normally distributed, but the bars in blue would indicate to you that responses to inoculation can be somewhat impressive, reaching 50% greater, perhaps even greater than 75% greater than the non-inoculated control. But of course, on the other side of the spectrum, we have yield being cut in half or reduced by 25% with inoculation. Lots of variability. We can plot those same I over NI values in what is called a piano key plot, simply arbitrarily numbering the plots from left to right, high to low in terms of response, and again, get a visual read on the distribution of the data in that way. Same way for fall 2000 lettuce, and you seeing the 2 different types of distributions within the same experiment. So again, just returning to the reference about multiple literature supporting the idea of using different statistical approaches depending on the hypotheses being tested. In our case, the candidates that we're currently focused on are listed in the lower left of the slide, and we're also asking the question about what role transformation may play in analyzing the many data points that we have. And finally, I'll mention mapping and the steps that we are taking to understand the distribution of our values by simply plotting, if you will, by locating within the plot map where the positive and negative plus greater than 1, less than 1 I over NI values occurred. And then asking the question, why might that distribution be the way it is? Is it, as it is shown here, somewhat lopsided with the greater than 1 values being towards the west end of the field area, whereas most of the less than 1 values are to the east? Or in this case, in a high tunnel, why are most of the green plots on the left side of the tunnel, the southern side of the tunnel, versus the eastern side of the tunnel? A step that I don't always take, but I am becoming much more comfortable and interested in taking and understanding the distribution of the data. So far, product application based on our standard analyses of variance and preliminary means testing, product application has rarely resulted in an increase in yield or change in quality. And so to summarize, we have covered a large amount of territory in terms of describing the 11 experiments, the 39 runs of them conducted over 3 years, and now embarking on a process whereby we look to understand the outcomes using different statistical procedures to complement the ones that we are very comfortable with and are very common within the product testing category. Again, thank you very much.